Hello everyone, welcome to Get My Class. So let's solve this question here. It says Bt toxin gene has been cloned from dash and been expressed in dash. Option A plants, bacteria. Option B bacteria plants. Option C bacteria. Option D plants. So let us understand the question. It says Bt toxin gene has been cloned from dash and been expressed in dash. So we have to tell that from where this Bt toxin gene has been cloned and where it has been further expressed. That's what is the question. So before we look for the correct option, so let us first talk about what do we mean by Bt toxin. Bt toxin. So Bt toxin is nothing but the toxin which is being, usually it is produced by a bacteria which is called as Bacillus thuringiensis. So B for Bacillus, and T is the, it is a particular strain of bacteria, thuringiensis. Okay, this is a type of bacteria which has the ability to produce certain proteins, which we call it as toxic protein, or you can say crystal proteins. Okay, these can produce crystal proteins, which will have, pesticidal or you can say insecticidal effect on certain group of organisms. So example, you could see lepidopterans. Example, there are the other group of organisms also which get killed because of this crystal protein which has been produced by Bacillus thuringiensis. Now these lepidopterans or other group of insects, they have what do? They are involved in spoiling the crop. So they can bring spoilage of crop, right? They can bring spoilage of crops. Okay. So to avoid this, what can we do? Since this bacteria has the capacity to produce toxic protein, that is nothing the crystal protein or the cry protein. Okay, what do? We can take this protein, and if we produce the same protein in plants, then what can happen? Then the plants will produce this crystal protein, and when these plants feed on the plants, I mean these insects feed on the plants will have it will have crystal proteins or the toxic proteins, and when they feed, when the lepidopteran feed on it, and they will die because these crystal proteins will be produced in the plant. It may not harm the plant, but it will harm the insect which feeds on it. And that is how we can make, insect resistant plant. Okay. So what do? We take the gene for this crystal protein. So we take the gene, okay, from this Bacillus thuringiensis, and this will be incorporated into plants or plant cells. Okay. Now, once the plant will grow, what will happen? This gene after cloning, like we will amplify this gene and then we will incorporate into the plants. Okay. And then these plants, like these cells of the plants, will produce Bt toxin. Okay. And when the insects feed on these plants or the cells of the plants, the Bt toxin will be active, and then further it will kill the insects. Okay, it has specific effect where these Bt toxin actually, this Bt toxin works on the gut cells of the lepidoptera. Okay, and that's how they die. So let us see. It says Bt toxin gene has been cloned from dash. Cloned from what? It is from Bacillus thuringiensis, right? And been expressed in plants. So let us see. Option A says plant. So it is not cloned from plants. So hence it is ruled out. Option B says bacteria. Yes, it is being cloned from a bacteria, that is Bacillus thuringiensis, and it is being expressed in plant. That is the correct option. Option C says bacteria and bacteria. Of course, it is cloned from bacteria but not expressed in bacteria, because we don't want the bacteria. We want the plant to be insect resistant, right? So that's why. Then option D is plant and plant, which is totally wrong. So what is the correct option? The correct option is option B. So I hope you understood the explanation. Thank you, everyone.